So today I'm going to talk about highly pathogenic avian influenza in colonial seabirds in the Netherlands and focusing on the terns. Because I'm a virologist by education and by profession, I cannot really resist introducing the virus itself. So influenza A viruses are classified by the major surface proteins. These are the neuraminidase and the hemagglutinin. These are on the outside of the virus particle. And this is where the abbreviation of the virus types are coming from. So the H and the N is actually these proteins on the outside of the virus. But you can also see it on this picture, these little particles here. This is the genomic material of the virus. What is very special about it, this is an RNA genome, so unlike the humans, they have RNA as a genome, which is very, very variable, so there are mutations occurring which are not being corrected. Plus, they are also located on 8 separate segments, and the viruses, when they infect a single host, 2 different types at the same time, if they infect the same duck, for instance, then these viruses can also mix with each other. And these 8 genetic segments can be exchanged, and all kinds of variations can occur. So this is actually the secret of their variation. And this phenomenon we call a reassertation. Low pathogenicity avian influenza strains can be found in the anseriform and in the caridiform birds. And sometimes with the genetic changes, the H7 and the H5 types can evolve into highly pathogenic strains, but only those 2 types. So this happened in 1996, as already Case mentioned, in China, in Guangdong province, in a domestic goose farm. And this was the first highly pathogenic avian influenza that managed to establish a sustained transmission between poultry. There were an outbreak in the 60s, actually already in common terns documented, which was also a highly pathogenic avian influenza strain, but luckily that one died out early on. But unfortunately, this one did not, and in 2005, the virus actually stepped into the wild bird populations and started to spread towards Europe, towards Africa, and actually it made a whole trip around the world. In 2005, there was an outbreak, and then there were also sporadic cases for 10 years. And then we see with the higher and higher magnitudes the virus appearing and reappearing in poultry and also in wild birds. And as I already told you, because of the genetic segment changes, these viruses can pick up different N, so neuraminidase segments. So that's what you can see here. Sometimes the H5 is combined with N6, sometimes with N8, sometimes with N1. So this is the explanation for this nomenclature that you can come across. So as of 2016 on, the H5N8 type was the dominant. Probably you remember it was quite dramatic in geese and ducks. It was causing high mortality in those species, but it was still showing a seasonal pattern. So when the geese and the ducks arrived in the autumn period, then we saw a peak in the winter time, and then for the summer the virus actually kind of disappeared from our continent. But unfortunately, from 2021 on, there was a variant appearing. It was an H5N1 type, and this lost the seasonal pattern. So basically, it was present all year around, also in the summer. This is what you can see here below on the graph. So we had first the H5N8 in geese and ducks, a little bit of, silence before the storm. And then H5N1 took over. And since then, we don't really have, we have a little bit of seasonality, but actually it's present here the whole year around. And then the gull virus, you also often see this expression nowadays. So I explained that there are a lot of different types of avian influenza, and based on their hemagglutinin gene, they can be classified into multiple groups. And the H13 and the H16 type, these are gull-specific variants, and they gull and tern-specific variants, and you cannot really find them in other bird species. And because they are also impacted, the gall species, by the highly pathogenic avian influenza H5N1. If they are infected with the two different types of virus at the same time, then reassortations can happen, so that you can have a very highly pathogenic and gall-specific virus as an output. Often people ask, like, oh, but we don't see anything now in poultry. Why is this? Why is this happening? Are they less susceptible these days? And actually, one of the possible explanations is that now this variant that is lingering around in the wild bird populations is not coming into contact with the poultry so often anymore, because the grote stern is a little bit less prone to visit poultry farms than, for example, a smint. Sorry, I don't know the English names of the birds, so I will just use the Dutch, but you'll understand, of course. Yes, but let's travel back in time to 2022. When the disaster happened, actually in the Netherlands, or when it started with the terns, the first reported cases were from May, and within 1.5 months we lost over 9,000 adults. These were the ones that were actually physically picked up, which is a little bit more than 20% of the adult population. But then if you look at the migration counts here below, actually the loss looked even more dramatic. It was approximately 50–60% of the calculations. But of course, we had to wait 1 year to see what comes back after the migration season. And as you can imagine, if you lose so many adults, then the breeding success was also close to zero. If you look at the map of Europe in 2022, you can see that the Netherlands was kind of the epicenter of the outbreak. The red parts in the diagram, those are the ones that were found dead. In Ireland, in the Scandinavian countries, in the Baltic countries, and also the northern France part was mostly spared. So here they didn't report mortalities. And if you look at the phylogeny of the sandwich tern viruses that were detected in 2022, I don't expect you to read this small font size here, but what is important is that the pink viruses are the ones that were isolated from sandwich terns. So what you can see here is that the virus was introduced into the sandwich terns in 2 different time points, so from 2 different sources probably. But then these 2 different clusters, they can also be found throughout the Netherlands from north to south, and also sometimes in the same colony you have both virus variants present. So the variety is really enormous. And this was also published in an article in 2022. But why the sandwich terns? Well, if you have seen a sandwich tern colony from close by, then you can probably answer it relatively easily yourself. They are really breeding close to each other, so it's like 30 centimeters, and they are lovely birds, but not the cleanest ones. So they are pooping all around the nests, and also arguing a lot with the neighbor, because they are of course very tight to each other, and they have to protect their little nest and little area that they have around their nest. So there is a lot of interaction, and they are also breeding together in mixed colonies with galls. So if the gall is bringing in a virus, they can easily interchange these virus variants between each other. Another thing with the sandwich terns is that they love visiting other colonies, especially the floaters, the non-breeder young birds. They are not settling in one colony, but based on the coloring readings, we know that in one day they can turn up in 2–3 different colonies, even further away from each other. So they are serving as excellent vector for a virus. Likely they are also highly susceptible for the variant that is circulating around, and they didn't encounter this virus before, so they are highly susceptible. They don't have pre-existing immunity. Unfortunately, common terns were not luckier either. This is one of the rafts in Zeeland. Actually, according to the original nest counts and the amount of birds that we found dead in the colony, we lost 90% of the adults breeding on that raft within 2 weeks. What we also saw that in the natural islands, when the birds are a little bit further away, further breeding from each other, they were much less impacted. Unfortunately, this pattern changed in 2022, which we couldn't really explain so far. And what is also interesting that in 2022, these outbreaks were mainly on the coastal colonies, and in 2023, it started to move inland, and also colonies in Hungary, for instance, were impacted by flu. So it really left the coastal areas of Europe. By the end of the breeding season in 2022, the numbers decreased. Well, kind of logical, because the birds are not concentrated so much around the colonies, so there are much less interaction between them. And they also started to migrate, of course, to the south, or at least what was left of them. And the other factor that we cannot really confirm, of course, is that if they disperse, even if they die, we cannot really find them, because in a colony you can go count, you know where to find them. But if you are checking the whole stretch of the African coast, for instance, or the European coast, that's really a difficult task to actually judge how many birds died after the breeding season. And yeah, they started to migrate to the south, which didn't look any better. In terms of outbreaks, so there were outbreaks reported from South African Republic, from Namibia, from different species, and from Senegal and the Gambia we have exact numbers, which is as exact as it can be, because they don't really have resources to do a survey on the entire coast. But what we know for sure, they collected over 10,000 carcasses of different terns, mainly West African crested terns and Caspian terns, but sandwich terns were also found among the victims. And then the springtime didn't look good either to welcome our migratory birds back in the Netherlands. We had outbreaks already as of January, February in mainly black-headed gulls on the roost sites, and then when they started to arrive in the colonies, sometimes the adults were already dead before they actually started breeding. But then the surprise came. So in the spring of 2023, we expected actually a low number of returning adults of sandwich terns. But compared to the magnitude of the disaster in 2022, the established breeding attempts were not even that low. So it was like a little bit over 15,000 nests that were counted in the Netherlands over all these colonies. I also calculated in the colony in Zeebrugge, because actually this is almost the Netherlands, or at least it's the same. I'm sorry, I hope there are no Belgians here. But, it's the same metapopulation. So basically the birds that abandoned the colonies, also in 2022 in the Netherlands, many of them ended up in Zeebrugge and trying to breed there instead of the disastrous Dutch colonies. Kees already mentioned the floaters. So those are the non-breeder birds that are there as a buffer in a population. And when available breeding sites will, when they become available, then these floaters will settle. And also in 2021 and 2020, we had a very good breeding success of sandwich terns. So these young adults likely returned now for the first time to Europe, and they filled up the gap that the old birds left behind the year before. And maybe you remember the map I showed before, that the Netherlands was the epicenter. So the other colonies in northwestern Europe will not hit that hard in 2022. So maybe those birds were recruited in our colonies. So that's why the impact didn't look as dramatic as we expected based on the losses in 2022. So we started with good numbers, and we also experienced minimal adult mortality throughout the year. It was less than 160 birds in total. So if we compare that to the 9,500 confirmed cases the year before, it's actually quite positive. But then just before the fledging, this happened. This is a picture from Zeebrugge. So several thousands of chicks had to be discarded. So the breeding success was on average around 0.3 in the Netherlands and Belgium together. We also have to mention the black-headed gulls. This is the Dutch Delta. In the Delta alone, we documented over 12,000 cases, so it's enormous. 20% of this was adult bird. And similarly to what we saw in the sandwich terns just before fledging, the outbreak peaked, and sometimes the breeding success in certain colonies was 0 because of the virus. So as you can imagine, if you have 12,000 birds dying within the Dutch Delta alone, then you have some management challenges to face. The colonies belong to different organizations, and often there is no consensus of what to do, which is kind of understandable. But decisions have to be made if they should clean or not the colonies and the public places, if they decide to clean, how to enter these colonies, if they collect the carcasses, where to deliver, because it's also not just a small amount, it's really several bags. And of course, there is very limited budget available for all this, for the work of the people, for buying PPE and so on. And we have already survived 2 years like this, and there are still several points that are not clear, actually, what we should do and how we should react. Case already mentioned the use of cleanups or possibility of cleanups. There are pros and cons, of course. Obviously, if you enter a colony, you will cause disturbance, so that's no question. And that unfortunately can also help to spread the disease throughout the colony, because the chicks are running away from you and they can interact more than they would normally do. However, if you leave all this outside in the colonies, then if the environment is as such, then the virus can survive for a longer time. And what is even a bigger problem is that the birds are interacting with the sick birds. This is a very strange phenomenon. It's called necrophilia. So the sick birds look like a female that is ready to mate, and then there are 12, 20 terns attacking this bird, interacting with it, of course, very intensely. So very dangerous in times of outbreaks. And they are, of course, also attacking these birds because they behave differently than the others, and they want to get them away from the colony. So they are picking on it and chasing it and so on. And what is also quite dangerous is that these carcasses serve as a food source for many different species, so including avian predators, rats, foxes, small carnivores, stray dogs. And I also put here human, because what you can see here, it's a quite small picture, but I took it in the Gambia last year. So actually on the beaches, people are collecting these carcasses and they make barbecue. So it's not just picking up or, oh, don't touch it, they actually eat it. And of course they cook it, but they have to clean it before, and they are not wearing gloves, I can tell. These are stray dogs also visiting villages after they had a meal on the beach. And this is actually a picture from Moart, Kenya, where we cleaned up several hundred gull carcasses, but actually the rats were faster, so the whole island looked like a pudding. So it's giving all excellent opportunities for mammalian transmission. And then, of course, we also have some scientific challenges, because if you don't care, just trash everything away, don't count, don't check the species, then we will have really hard time to estimate what is the actual impact on the different populations. And public beaches are often cleaned up with volunteers, and they are not educated. What is the difference between a Mediterranean gull and a black-headed gull? And they also don't care about rings, which, I mean, I don't blame anyone, but it's just how it goes. And of course, it's also causing quite big loss for the scientific projects. But we can also learn about avian influenza, because if we don't know the enemy, we cannot know how to deal with it. There is a very nice initiative led by the Vogeltrekstation and Erasmus Medical Center, the virus science team. This project is called the Zoonosis Project. Probably many of you are familiar with this initiative. In the frame of this project, the ringers can collect samples for avian influenza studies. We also joined this initiative, and we collected over 500 samples from the data, and we were focusing, of course, on the gulls and the terns. So the swab samples are collected to check for the actively infected birds. I put back a virus picture to explain, because I see sometimes confusions among the birders. So when you collect the swab samples, these are sent into the laboratory, and they will isolate the RNA. So they will isolate this part of the virus, and they will target it with the PCR method. Since corona, probably everybody knows what PCR is, but it's a very targeted molecular method. And if they find that there is influenza in this sample, and if there is enough virus in the sample, then they will also try to sequence it, and then they will identify the entire genomic sequence of the virus, and then they can tell whether it's a highly pathogenic strain or not. We also take blood samples. These are separated by centrifugation, from the whole blood. And the serum contains the antibodies. So actually, when we are doing this, then we are not detecting anything from the virus. We are detecting the antibodies that are developed by the bird's immune response. So you can find these molecules in the blood, and we are looking for those. If you find them, that means that the bird has been infected before with avian flu. And of course, we also coloring each bird, so we can trace their longer-term survival. And you have to be careful how you work, of course, because you don't want to get infected yourself. We are not susceptible for the virus per se, but you never know. If a new variant comes up, then we might be, or we don't want to be patient zero. And we also have to make sure that we are not spreading the virus from bird to bird. So we are wearing gloves and disinfectants between each handling, and also the tools, so that you are not the one who is putting the virus from one bird to another. So from the sandwich terns, we performed a quite big sampling campaign. We had 4 sampling locations in total, one on Texel and the Prins Hendriksandijk. This sampling was performed by Mardek Leopold and his team, and they sent the samples to Lelystad, to the Bioveterinary Research Center, and all the rest of the samples were collected in the frame of the zoonosis project, and we also collected samples from Zeebrugge for the reason I already told you. It's the same metapopulation, so we also wanted to take this along. The adults were caught by walk-in traps, and the chicks we sampled when they were approximately 15 to 25 days old, so they were just actually about to fledge. We sampled almost 150 adults and almost 180 chicks in total. And some results. So first, have a look at the adult birds. We had a very low number of adults carrying the disease at the time of sampling. Actually, on Texel, 0 birds that tested positive out of the 42 sampled. In Zeebrugge, we found a single bird, and on Bleak, 3 out of the 45. Well, one of the explanations for this is that we sampled the birds just when they came back from migration, so those were probably the fittest birds that made it, that didn't die in the wintering areas, and they also had less interaction yet because they just started to settle in the colonies. And 2 out of these positively tested birds also survived. Also interesting to know that these birds had not showed any antibodies yet at the time of sampling. So they were positive, they had no antibodies yet, so it was a fresh infection, and they managed to survive, you know that based on the coloring readings. And there was a 3rd bird out of the 4 positive, which we found 1 month later with the clinical signs. So it's really interesting because we thought that after the infection, the clinical signs will appear in a few days. N is 1, so I cannot really detail further, but it was an interesting encounter. What we saw in terms of seropositivity is actually a difference between the colonies, which we couldn't explain. in the Zeebrugge site, we had 25%, followed by Bleak with 44%, and then 56%. The highest seropositive was on Texel. We don't know the reason why this difference is there, but it's good news that at least we have seropositive birds, and that's probably also explaining why we saw much less mortality this year in the adult population. Regarding the chicks, so it's good to know that the female birds can transfer the antibodies through the egg yolk to the chicks, and they will be protected for the first couple of days of their lives. In chicken studies, they showed that these are not detectable anymore after a week or so. While with the sandwich tons, we didn't want to sample the chicks so early, at such an early age. So at the time when we sampled them at 15 to 25 days old of 25 days, they didn't show any maternal antibodies anymore. We only found 4 seropositive birds, but they were also actively infected, and that means that they were already mounting their own immune response, so probably that was the reason why we found them positive. Regarding the active infections, we found that on Bleak, the highest number of chicks were found positive. Actually, here we had multiple sampling points, and we also had one very late in the season. That's probably why we detected so many positive ones, because at the time of fledging, the virus was taking over, and likely that's skewing the numbers a little bit, because the mortality was equally high everywhere at the end. So actually, if we sample all the colonies at the same time of the ongoing outbreak, then we should have seen more or less the same picture. All the samples that we collected and were analyzed were positive for highly pathogenic avian influenza. It was confirmed from all the samples. And interestingly, on Texel, we only had one positive chick at the time of sampling, and this one chick was also found back later alive. So there is a glimmer of hope. I will have the last slide about this occurrence. So I selected a few. We have so far 5 chicks that were testing positive out of the 42, that survived out of the 42 that were testing positive. This one was a very recent one. Red J43 was one of the chicks out of the 4. It was seropositive, so it was already carrying antibodies, and it was also actively infected. So actually we just caught it at the time when it was fighting against the disease. And when they fledged, of course, we do a very intense ring reading activity on the Dutch coast, but we cannot see everything. And our colleagues in the Gambia reported this bird a few weeks ago, and it's happy and alive. Blue TZR is the chick that was tested positive on Texel. And actually this bird I found back myself at the Thijshorst in Portugal on a ring reading expedition. And then later on the bird turned up on Menorca, and it's spending the winter there, still there. I get weekly reports from the Menorcan people, and they are super happy that they have such a special bird on their coasts. This was the bird that I mentioned before. It's actually quite a blurry picture, but it was very far. And with ring reading, you cannot always select your conditions. So this bird was tested positive on Bleak at the time of sampling as a breeding adult. And then 1 month later, I found it with the clinical signs. So this was actually the time when it was, but it was still able to fly. So we still have some chance that we will find it back one day. It would be a very nice record. So to summarize, we have 2 consecutive very poor breeding seasons behind. And unfortunately, we didn't only lose chicks, but also very valuable adults from the population. The long-term consequences are still unclear. So we always have to see, let's say, the next breeding season, how it looks, how many birds are coming back, because we have a big black hole, let's say Africa. We don't know what's happening there. And also what Case mentioned before, we don't know how many buffer we still have left. So the floaters that were present in the healthy population and that managed to fill up the gap more or less after the first big outbreak here, they are not there anymore. So if we get another year like this or another hit, we don't know what is going to happen. So how big the drop will be after that. For now, it looked okay in the sandwich terms at least, but I'm not sure that the next wave would be so easy to survive again for the population. We saw a very good number of sandwich terms already showing antibodies, but we don't know much about these. So it's nice to know that it's positive, but for how long will that stay like that? So the birds that we found seropositive, they were in the adults, they were not actively infected. That means that they mounted that immune response either in the previous season, that's good news because it's there for 1 year then, or they mounted it in the wintering areas, which is also like a few months at least. But whether these antibodies are protecting them in the next outbreak when a new variant comes, that's also a good question, we don't know. So in the chicks, yeah, it's another topic, of course, because they always hatch with the naive immune system or more or less naive immune system. They get antibodies from their parents, but for how long they are protecting them, we also don't know. So there were speculations or suspicions that if they get maternal antibodies, that will protect them. But maybe that's why we saw the high chick mortality when these maternal antibodies are fading away. They are not ready yet to actually mount their own immune response and survive. But because we have the survival chicks, there is still chance. So we have to just go sample and try to learn as much as possible about this. And what can we do? Well, it's unfortunately quite limited. And there are no cure available for this. And also, in terms of vaccines, there are some experimental ones for the poultry industry. But many of these will be, if at all, very, very difficult to use in a wildlife setting. So that's, I don't think we should rely on it for now, at least. Cleaning up the carcasses, well, in the Netherlands, we are quite flat. So at least the colonies are approachable. Whether it's a good idea to clean out or not, this is still also subject of discussion. Because if we can catch an outbreak early enough, there are papers that demonstrated that it's useful so that you can limit the magnitude of the outbreak, but we have really limited data to actually say something really sure. What we can do, well, nothing new there. We have to provide enough breeding areas for them. With the terns, we have to be also careful because what we saw on the rafts, especially in the first year of the outbreak with the common terns, we must make sure if we provide artificial rafts for the birds, that these are not serving as ecological traps, because these are squeezing the terns a little bit closer to each other than in a normal natural island situation, and then because of that the transmission rate of the virus can be higher than on a natural island. Also regarding the undisturbed roosting sites, because of the recreation activities in the Netherlands, there are really limited undisturbed beaches, and also in the industrial area of the Maasvlakte, the biggest tern roost site was just ruined last year. So yeah, they have to go on the public beaches again. Oh yeah, and we also have to ensure that the predation stays low, not only to protect the birds, but also to limit the level of or the chance of mammalian transmission of the virus. And of course, we have to learn as much as possible about the virus. And there are a lot of people behind all this work. I think it moved each segment of the ornithology community of the Netherlands and also Europe-wide. So very big thanks to everyone who is working on this, and also to all the ring readers, not only in Europe, but also in Africa. And all the organizations behind. And a little bit of advertisement. So we are writing every year a report for the province of Zeeland about the mortalities that are occurring in the south part of the Netherlands, and unfortunately in the last 2 years the bird flu is one of the main topics. So if you're interested, it's available in Dutch and in English as well. Or actually not yet, but next week it will be published. Thank you for your attention.